Human cloning and ethical issues of transgenic animals. Human cloning is the creation of a genetically identical copy of a human. The term is generally used to refer to artificial human cloning, which is the reproduction of human cells and tissue. The debates on cloning generally revolve around moral, ethical, and religious arguments. These ethical concerns have prompted several nations to pass laws regarding human cloning and its legality. Two commonly discussed types of theoretical human cloning are therapeutic cloning and reproductive cloning. First one, therapeutic cloning. Therapeutic cloning would involve cloning cells from a human for use in medicine and transplants. It is an active area of research but is not in medical practice anywhere in the world. Two common methods of therapeutic cloning that are being researched are somatic cell nuclear transfer and pluripotent stem cell induction. In this, the growth of the embryo is stopped at the blastocyst stage. Next, reproductive cloning. Reproductive cloning would involve making an entire cloned human instead of just specific cells or tissues. In this, the cloned embryo is transferred into the womb of a surrogate mother. Ethical issues of transgenic animals. Many ethical concerns have been raised about transgenic animals' potential harm to human health, potential damage to the environment, excessive corporate dominance, and the unnaturalness of the technology. If the genetic manipulation of living organisms by humans goes unregulated, it will not only lead to the exploitation of the organisms but will also have harmful impacts on our ecosystem. It is wrong at the moral and biological level. This is why we need ethical standards to regulate the human manipulation of organisms. In terms of genetically modified organisms or GMO, the Indian government has set up the Genetic Engineering Approval Committee. This organization makes decisions regarding the validity of research involving genetically modified organisms and addresses the safety of genetically modified organisms introduced for public use. One aim of genetic engineering is to increase the growth rate and yield of animals like cattle, pigs, and poultry. There is particular concern about the way we exploit animals for food and the development of medical products. Medical experiments may involve a certain amount of animal suffering. An example is provided by the on-cow mouse, which was the first animal to be patented in the US. The on-cow mouse is a transgenic mouse to which an on-cow gene has been added. Some people argue that patenting animals is itself unethical because it reduces them to the level of objects. Others argue that experiments such as those with on-cow mice cause suffering and should therefore be banned. At the other extreme of animal welfare and those animals that are used for pharmaceutical products such as AAT and factor IX, which are probably the best cared for farm animals in the world because they are the most valuable. We know that in the early 1990s, the informed opinion said the same thing about cloning from adult cells, but Dolly arrived in 1996. Thank you.